Hi, this deck of cards was compiled to facilitate working through the MCAT-style passage, diminished sensory input, and the enjoyment of food. Pheromones bind to receptors in the blank olfactory epithelium, while standard odor molecules bind to receptors in the olfactory epithelium. So what type of receptors do pheromones bind to? Those things that, I don't know, like dogs can smell really well and we're supposed to be attracted to them in the opposite sex. So what's the binding site? It would be the accessory olfactory epithelium. And this animation is like a janky but a good rendition of what the accessory olfactory epithelium layout is like. There goes the nasal passage. Okay, and these are axonal projections. This region would be your vomeronasal system. Okay, so just watch and learn. Just take it in. You have your apical and your basal zones. And this would be like a pheromone binding, this little green triangle, sending a projection to your accessory olfactory cortex in your brain so that you can process what's being smelled. Okay? G blank I are regions within the olfactory bulb that receive input from olfactory sensory neurons sensitive to a particular odor. So which regions within the olfactory bulb receive the sensory neurons? I'll give you a moment to think. This topic really gets down to the microscopic level. That would be the glomeruli, and here you can see the glomeruli receiving projection signals from, your olfactory epithelium. After they fire action potentials, you can see them being fired into the glomeruli region. Okay. Odorant receptors are G protein coupled receptors, that is GPCRs, that open blank gated channels. Blank gated ion channels. So what molecule is associated with these ion channels? I'll give you a moment. That is cyclic AMP, and here goes an animation of the formation of cyclic AMP from ATP. It's cyclic adenosine monophosphate, okay? So when these little things bind, like to ion channels that would be near the protein membrane, they would open. But when they dissociate, they're gonna close. There's another video that I have that shows that in like the rod of the eyes and the retina. So you should probably find that, and then there's a card in there somewhere about cyclic AMP being binded to potassium channels, sodium and potassium channels. Okay. Odorant receptors, again, are G protein coupled receptors, GPCRs, that activate adenylate cyclase to elicit various blank-dependent protein kinases. So by now you should know the answer. I'll give you a big hint. We just looked at it. So what's the answer? So again, the answer is cyclic AMP. And we see the adenyl cyclase, the big brown doodle-looking thing just now, the enzyme that forms cyclic AMP from ATP. And then cyclic AMP are the little red dots associating with this protein kinase here, okay? Which is then going to elicit a cascade of other events. So again, adenyl cyclase, making the little circles, see cyclic AMP. Then cyclic AMP binds to protein kinases, which then elicit other various responses in a cell. A control sample is necessary in order to measure, is it a baseline or the maximum response in a population? Which answer is it? And the answer is baseline. So let's say you have a population of students and you're testing, the amount that drinks water, the amount of caffeine usage. A baseline control sample would be like giving one group of students water, as pictured, and giving the experimental group, the caffeine. So the control group or the baseline response would receive water. So that's like an example for more clarity, okay?